Vesicle trafficking is fundamental to distribute correctly lipids and proteins among cellular compartments. A complex network of interconnected pathways transport vesicles between organelles to satisfy the cellular demand. Multisubunit tethering complexes MTCs form a group of 9 protein assemblies that are conserved from yeast to human and that are essential for intercellular trafficking. Exocyst, SL1, COG, GARB, HOPS, COLA, TRAP1, TRAP2, and TRAP3. Each MTC recognizes a specific type of vesicle and tether it to the appropriate receptor membrane, providing specificity and directionality to the trafficking. My research aimed to do a comparative analysis among all MTCs to shed light on the mechanism of action on these complexes. My group have developed a PITC protein interaction from imaging of complexes after translocation, a high sensitivity technique that allows us to study interactions between 2 proteins using live cell imaging. We first conducted in silico genome-wide screening to identify those proteins that could be functionally related to MTCs. We then used PIC for the protein interaction screen with the protein selected in the silico analysis. Given the high sensitivity of the assay and that the experiments were done directly in living cells, this method was prompt to detect interactions that have been overlooked by previous analysis, such as indirect, transient, or low abundant interactions. I found that MTCs establish a network of interactions with P480 passes lipid flapases that translocate phospholipids to the cytosolic leaflet of the membranes. P480 passes maintain the asymmetric distribution of phospholipids, and they also play essential roles in the vesicle trafficking. In yeast, there are 5 P480 passes: DNF1, DNF2, DNF2, DRS2, and NEOL. DNF1 and DRS2 are the ones that concentrate most of the interactions with the MTCs, mainly GARP and the traps. Up to date, no study has observed that MTCs and P480 passes might function together. In this thesis, I investigated the molecular mechanism that mediates the interplay in this network of essential components for the trafficking machinery. I found that GARP is responsible to transport DNF1 from late enzymes to trans-Golgi network during recycling of DNF1 to the plasma membrane. A transport step that is essential to maintain the correct distribution of PE in the plasma membrane. Furthermore, I found that DRS2 is required for the proper functioning of cytoplasm to vacuole targeting. CVT pathway, a model historically used to study selective autophagy. When the CVT pathway is triggered, both the TRAP3 and transmembrane protein named ATG9 reach the phagophore assembly site PAS to start the process. The results of my thesis show that in response to temperature decreases, the cell induces the binding between the TRAP3 and the DRS2, that this interaction is required to sustain the adequate transportation of ATG9 to the PAS. In line with this, I have seen DRS2 is fundamental for the biogenesis of vesicles loaded with the ATG9 from the endocytic compartments. Altogether, this data evidences a new function to this flippase in the CVT pathway that might have important implications in other types of autophagy. Additionally, I identified a short motif in the N-terminal tail of DRS2 that is required for the interaction TRAP3-DRS2. This is a motif highly conserved among P480 passes and other proteins that bind MTCs. What suggests that it might have a more general role in regulating the trafficking of vesicles that remains to be elucidated.